Bees pollinate many wild plants and crops and are therefore essential for human well-being. To effectively protect bees, the effects of different environmental stressors can be assessed in field and semi-field experiments. While past research focused mainly on social bees, as honeybees or bumblebees, for the assessment of stressors, there is increasing interest to study also solitary bees. This is a semi-field experiment that investigates the effect of a pesticide on the cavity-nesting solitary bee species Osmia Bicornis. Before bees are released into the flight cages, they are hatched under standardized conditions. Because individual bees shall be tracked in this experiment, we mark them with unique IDs that are produced for honey bee queen rearing. The nesting unit used for this experiment offers about 5 nest cavities per released female bee. The nesting unit is composed of wooden layers which can be assembled to a block, each layer containing 10 nesting cavities. The cavities are semicircular and open at the top, which allows to look inside the nest and observe nesting progress and offspring development. A plastic foil covers the layers and protects the nesting females and brood from disturbances. To follow the larval development until spinning of the cocoons, we regularly check the nests and record survival and developmental times across instars. To document nesting progress by females, the most recent brood cell can be marked in each nesting cavity before and after the time span of interest. In this experiment, nesting progress is recorded every day during 3 weeks. Even the completeness of the cell can be noted down. We used values of 1/2 for incomplete cells that only contain pollen. A value of 1 represents cells that were completed and include also an egg and a cell wall made of mud. Nesting females are usually roosting inside the nesting unit during the night. We therefore take pictures from all layers after sunset to count the number of roosting females as a proxy for the number of female bees that are still alive on a particular day of the experiment. Impact of pesticides on bees may be modulated by other environmental stresses, such as the extent by which bees are affected by their natural enemies. In one of our field experiments, we therefore study how pesticides interact with impacts of parasites, parasitoids, and predators. A common parasite that can be found at our study sites is Caccacinus indicator, which belongs to the fruit flies and can be recognized in the bee's nest by its orange, hair-like feces. The larvae of this parasite feed on the pollen provisions in the brood cell and do not attack the larva directly. The bees developing inside attacked brood cells therefore often survive but reach a much smaller body size. Another common natural enemy of Osmia bees is the predatory beetle Tricodes Apiarius. Its larvae can be recognized by the pink color and large size. They predate on the bee brood and can destroy several cells and whole nests. The bombyliid fly anthrax is a common parasitoid attacking Osmia offspring but after the spinning of the cocoon. It therefore can best be detected when it hatches from the cocoon in the next spring.